Hey there, folks. Let's talk a little bit about cells. Welcome to Chapter 4. This is the first video for Chapter 4. When we dive into cell biology, we're going to be doing a lot of microscope work. And cell biologists use three major types of microscopes. Scanning electron microscopes are for getting high-definition images of the outside of cells, so we can see what texture they have, maybe what sorts of molecules they are using on their exterior to receive molecules and to communicate with other cells. Transmission electron microscopes give us very detailed images of a cell's internal structure. And the last type of microscope you folks are actually going to be familiar with through your lab work if you aren't already. These are light microscopes. In fact, if you've had lab already this week, those animal cells on the far right might look awfully familiar. They're prepared the exact same way you are going to prepare them in lab. Cells are extremely small, and there's actually an advantage to that. They do so much communicating with other cells by sending molecules outside to other cells and by receiving molecules at the surface, that being small and therefore having a very, very high surface area to volume ratio means that substances and molecules that are going to be sent outside the cell can get to the outside of the cell fairly quickly. When they get to the cellular membrane that wraps around the cell, there are specific places like protein channels where it is possible for large molecules to make their way into and out of the cell, like a gate in a fence, basically. The membrane itself can also be passed straight through by small non-polar molecules. This is because the membrane is what we call semi-permeable. Semi means part way. Permeable means that some things can pass through a substance. So a semi-permeable membrane can be passed through by certain things. In this case, small non-polar substances like water. Hey, water's important. We know this. The structure of that semi-permeable membrane is important all on its own. It's made up of two layers of lipids. We call it a bilipid membrane, a bilayer lipid membrane, because of those two layers. One end of each lipid can attract and attach to water, and the other one cannot. The end that can attach to water is called hydrophilic, meaning water-loving. The end that can't is called hydrophobic, or afraid of water. These lipids are arranged so that the hydrophilic ends are on the inside and the hydrophobic ends are on the outside, facing outside and toward the inside of the cell. This prevents water from binding to the membrane itself. If water was able to bind to the membrane, the whole membrane would dissolve, and there would go life as we know it on this planet. There are two major types of cells on this planet: prokaryotes and eukaryotes. Prokaryotes, of which this is one example, tend to be smaller and simpler than eukaryotes. You may see them with fimbriae or pili on their exterior. Those are little finger-like projections. They may have a long tail-like flagellum, and these two different types of projections are how they can move through the water or through whatever medium they are in. On the inside, on the inside of the capsule, you may find a cell wall and cell membrane and chromosomal DNA, which they locate in a region called a nucleoid, where the DNA is just floating inside the cell. Also inside the cell are very small organelles called ribosomes that make proteins, specifically amino acids, for the cell under the direction of the DNA. In the case of eukaryotic cells, as you can see, their world is a good bit more complicated. Eukaryotes are larger, for one thing. and they have many more types of organelles inside them, some of which that do highly specific jobs. There are two different types of eukaryotic cells: plant cells, which you'll see on the left, and animal cells here on the right. In both cases, instead of locating DNA in a nucleoid region of the cell, our DNA, yes, we're made up of eukaryotic cells too, our DNA is located in a specialized type of organelle called the nucleus, which you can see in that large purple organelle on the right side of either cell. Eukaryotic cells can be very specialized. In the case of cell walls, they have both a cell membrane, and in the case of plant cells, they have both a cell wall and a cell membrane to protect them. Animal cells only have a cell membrane. There are a number of other things that are different between plant and animal cells. Plant cells also have large vacuoles, which you'll see is that very large organelle on the left-hand side of the plant cell. And that is the storage organelle for plant cells. Plant cells photosynthesize. In fact, the place where photosynthesis happens is an organelle. It's that green organelle at the top and bottom of the cell, although in an actual cell you can find them anywhere, called a chloroplast. Chloroplasts, when they photosynthesize, make sugars, and at least some of those sugars are going to need to be stored inside a cell, at which point they get stored inside a vacuole. The vast majority of all organelles are actually the same between plant and animal cells, even though plants and animals are very different. Both plants and animals have a cytoskeleton. A cytoskeleton is made up of various tubes and fibers made out of protein that are floating inside the cell, and the organelles can attach to the cytoskeleton so they can be moved around the cell wherever the cell needs them to be. Peroxisomes are small organelles that contain acid and are used to break down either waste products or compounds that are going to be used for food, for energy. Mitochondria, singular is mitochondrion, are specialized cells that also help break down components for energy, and they are particularly interesting. Both mitochondria and chloroplasts over in the plant cells, we think were at one time actually separate organisms. Check that one out. In fact, they still have their own DNA, and mitochondria breed themselves, they replicate and split separately from the rest of our cells, which is pretty darn interesting. They maintain some independence, even though they've been part of our cells for millions of years. Ribosomes, next one down, are even smaller than peroxisomes, and they help make amino acids for the body under the direction of the genetic code that's stored in our DNA. We have two different types of endoplasmic reticulum, rough and smooth, two different flavors. Rough ER, as you'll see it called for shorthand, is studied with ribosomes, and rough ER also helps make proteins. Smooth ER helps make other compounds, like lipids. The Golgi apparatus helps make transport vessels. It sort of manufactures other little carrying organelles that can carry substances to the outside of the cell. So a little transport packet can bud off of the Golgi apparatus. It can take in whatever substance, whatever molecules needs to be carried to the outside of the cell, and it will connect to the cytoskeleton and slide its way through the cell until the membrane of the packet can fuse with the bilayer lipid membrane and release its contents out into the outside of the cell. Lysosomes are another type of organelle that are used for breaking out and digesting or destroying small amounts of other components. And last but not least, we have the plasma membrane. Again, animal cells have a plasma membrane, but so do plant cells. It's just that the plant cell also has a cell wall that is so much thicker than the plasma membrane that it's very difficult to see the membrane smushed up against the cell wall. Because the cell wall in plants is so thick, it can be difficult for plant cells to communicate with each other if it weren't for specialized structures like gateways that they have inside the cell wall. These are called plasmodesmata, and they are specialized places where molecules can get into and out of a plant cell, bypassing how thick, compared to the rest of the cell, how thick that cell wall is. Last but not least, over the far right, you'll see centrioles. Now, centrioles do appear in both plant and animal cells, even though here they're only labeled in animal cells. Centrioles help direct the cytoskeleton, and they play a big role in mitosis, which is the process where animal cells and plant cells reproduce themselves, at which point centrioles direct the cytoskeleton to connect to copies of DNA, to arrange them and get them positioned correctly so that each new cell has the correct amount of DNA and the right number of copies of specific genes of DNA. That way, when each new cell splits off, they'll both be able to function. Like I was saying before, let's go into this just one more time. Endosymbiosis is the technical term for what evolutionary and cell biologists know was the original relationship that our cells had with some bacteria. Over time, those bacteria became our mitochondria and our chloroplasts. They are organelles. They have been living in our cells for so long that they have ceased to exist completely independently of our cells. But they're still a little independent. They still maintain their own strands of DNA, and they still reproduce themselves separately from the schedule the rest of our cells follow. If you want a true example of utter geekiness, when biology geeks gather, occasionally someone will ask, so, what's your favorite organelle? Even worse, if you were an organelle, which one would you be? I would have to say, I would definitely be a mitochondria. I love chloroplasts. They are where photosynthesis happens, but mitochondria are the powerhouse of the cell. Yeah, gotta love